Uh, Period 1, Biology, Dvir and Walter.
The nitrogen cycle is very interesting, along with the process of a sunflower can be very exciting. It all starts in the soil, which is where nitrogen originates, so the plant can take it in and grow big and strong to produce seed. The plant uses this nitrogen to form other compounds. Thanks to bacteria, there's no fooling around because the nitrogen becomes usable. The plants suck it from the ground. The consumer then comes around, eats the plant, and retrieves it back into the ground, along with organic matter that decomposes and returns to the soil. Then bacteria in the soil breaks down the nitrogen so that it can return to the air and everywhere.
Without the nitrogen cycle, DNA would be useless due to all of its great uses. It's also known as deoxyribonucleic acid and is said to contain many of the traces of life. It's one of the nucleic acids information containing molecules in the cell and is considered and is in the nucleus of every human cell. The information in DNA guides the cell making new proteins in order to pass down traits from generation to generation.
Without the nitrogen cycle in DNA, mitosis would not exist. Without mitosis, neither of these would exist. Mitosis is a vital process where a cell duplicates into two daughter cells and includes 5 steps in order for these 5 steps to duplicate. It starts with interphase 1, G1 and G2. Here the chromosomes aren't easily visible because the chromosomes are uncoiled and the cell goes through normal functions of living and existing. After interphase comes infinite prophase, where the chromosome begins to coil and the spindle apparatus begins to form as centrosomes move apart. After prophase comes metaphase, where the chromosomes become aligned on the plane. Don't forget anaphase, where the chromosomes separate and then immediately proceeds on to telophase. This is the famous phase where the nuclear membrane reappears. The chromosomes uncoil and the spindle apparatus breaks down, which makes the cell divide into 2, creating 2 new cells.
Can't have mitosis without meiosis, and without DNA, life is useless. Meiosis goes hand in hand with mitosis, meiosis, prophase, metaphase, anaphase, telophase, nucleokinesis. It starts with prophase here in the first place. This is where homologous and chromosomes become paired along with chromosomal crossing over. It's definitely not over cause it has only just begun. So let's continue on to metaphase 1, where homologous pairs became aligned with in the center of the cell. Anaphase is where the chromosomes separate along with telophase, where nucleus of one cell divides into 2 nuclei. Finally, there is interkinesis, which is where the interphase, but with no DNA synthesis.
The replication creates species which get naturally selected and evolve from common ancestors due to genetic mutations which occur in DNA, which is RNA. Both get replicated through transcription. Certain mutations can be beneficial if it gives an animal an advantage. Then it becomes official, like webbed feet or different bone structure. This animal will live better and pass down its genetics due to an improvement in fitness. All living animals today are witnesses to this. This is called natural selection and is vital for an animal's life and protection.
DNA replication is the final destination because this is the main function of the whole operation. Replication, transcription, and translation. The DNA functions in the 3 main types. It starts with transcription, information. This transcription takes in 2 main steps, all of which takes place in the nucleus, starting with the formation of pre-messenger RNA with the involvement of RNA polymerase enzymes. It is finished in no time. The pre-messenger RNA is then changed to produce the desired mRNA molecules and multiplication. This is because on each occasion, when cell divides, each of its double strands of DNA splits into 2 single strands of DNA. Each of these single strands act as a template for new strands of complementary DNA. As a result, each new cell has its own complete genome.
The mRNA formed in transcription is transported out of the nucleus into the cytoplasm, to the ribosome, which is the home of the cell's protein synthesis factory. Here it directs protein synthesis. mRNA isn't directly involved for this, but tRNA has controlled the sequences. The process by which mRNA directs protein synthesis with the assistance of tRNA is called translation. Ribosome is the process's home.
Suck it, Griff.